All right, welcome to the third and hopefully final part of Chapter 5, in which we talk about membrane proteins and some of the concepts we need to know in order to understand their function. So, proteins embedded in the cellular membrane or the plasma membrane of a cell determine the functions of that cell's membrane. So it is all about proteins. Now, since this is not a majors biology course, let's talk about only two examples of membrane protein functions. Membrane proteins can be enzymatic, which means there are enzymes embedded in the membrane, and they can catalyze certain reactions. So you can have enzymes on the protein membrane helping out with chemical reactions required to keep the cell alive. Or, one of the most important types of proteins embedded in the membrane, the transport proteins. And transport proteins are involved solely in movement of molecules across the membrane. So, when we want to talk about movement of molecules, we need to talk about diffusion and osmosis. So diffusion is the random movement of particles from areas of high concentration to areas of low concentration. So here's my awesome German German diagram of diffusion. So here are all these particles here. And here's a membrane that these particles can move across. And they are highly concentrated on this side of the membrane and have a very low concentration on this side. And diffusion is the random movement of particles from areas of high concentration to areas of, low concentration. So, over time, these particles are going to randomly move back and forth across this membrane until they have equilibrium concentrations. So, diffusion continues until the concentrations of particles are at equilibrium. It's an important fact to remember. So, because this is a German diagram, I'll just go ahead and say, here is my semi-permeable membrane. Right here, these are my particles in a very high concentration. Here they are in the low concentration. And then over there, the concentrations equalize via the process of diffusion. Yeah. Okay, I apologize to any Germans out there for that hideous rendition of movie German accents. So, here is diffusion in action. So this is going to be ink in water. And it's a great example of how particles move randomly in their movement from areas of high concentration to low concentration. Or not. I demand it. Here we go. So the ink is entering the water, and you're going to see all these little tendrils move in random directions. See that? And sometimes large amounts, we had it highly concentrated here, and they moved around, and suddenly it's highly concentrated here still, even though it's moving around. And then over here we've got things moving in random directions. But over time, this is still moving towards even concentrations throughout the entire container. So it's moving towards equilibrium, even though you can see that sometimes they move into high concentrations in random places and then diffuse out from there. It's random movement of particles from areas of high concentration to areas of low concentration. There it is in red, because it's pretty in red. Anyway, moving on. Okay, so when we talk about diffusion, it's important to talk about the concept of the concentration gradient. Concentration gradients are a way of describing the behavior of diffusion, or describing the aspect of concentrations. So, when a particle moves along its concentration gradient or down its concentration gradient, it is moving from areas of high concentration towards areas of low concentration in order to reach equilibrium concentrations. So concentration gradient describes, the high to low. So that moving along the high to low concentration gradient means moving from areas of high concentration to areas of low concentration, which is diffusion. So, an particle moving down or even along its concentration gradient is moving from areas of high concentration to low concentration. Hopefully, that was repetitive enough for you to remember it. All right. So diffusion is random movement of particles. Osmosis is the diffusion of water. And in biology, when we talk about osmosis, we talk about the diffusion of water across a membrane. So let's talk about some terms that we're going to use when we talk about osmosis. And those are isotonic, hypertonic, and hypotonic. In an isotonic solution, you have the same concentration of water inside the cell as outside the cell. That means that there's going to be no sort of concentration gradient to move along per se. Water will randomly move across the membrane back and forth, but every time some water enters the membrane, you create a concentration gradient, so water exits the membrane. So what you have is, no net movement of water across the membrane. Think of it like me trading money with my friend. And an isotonic trade, I'm giving my buddy $5, and every time I give him $5, he immediately gives me $5 back. Then I give him $5, and he gives me $5 back. Is there movement of money? Yes. I'm giving $5 to him, and then he's giving it back to me. But is there any net change in the amount of money I have? No, because I still am having $5 at pretty much all times. So there's no net movement of water across that membrane. Now, when we talk about osmosis, I'm simplifying it in order to talk about the concentrations of water. So I'll often refer to these tonicities, these isotonic, hypotonic, hypertonic, in terms of the concentration of water inside the membrane versus outside the membrane. But really, the concentration of water inside the membrane versus outside the membrane is governed by solutes within the cell, within the membrane, that cannot cross that membrane. So normally, if I had a high concentration of solutes inside that cell, we'll say like molecules of starch, right? Starch is too big to easily cross a membrane. So if I had molecules of starch inside the cell, and there are ways for starch to cross a membrane through transport or endocytosis, but we're not talking about that. We're just even going to just mention just a membrane. So a membrane, a cell-like membrane. If I have a bunch of starch inside there that can't cross the membrane, then I have a high concentration of starch inside the cell and a low concentration of starch outside the cell. But that starch can't diffuse. So the only way to even the concentrations is to reduce the concentration of starch by increasing the amount of water inside that cell. So say outside the cell there's like 4 starch molecules per milliliter. And then inside the cell there's like 30 starch molecules per milliliter of water. So in order to even the concentration inside the cell versus outside the cell, I have to add water. So if there's 4 molecules per milliliter outside the cell, and inside there's 20 molecules per milliliter, I am going to jack up the amount of water in that cell until I can get the fraction that equals out 4 molecules per milliliter. So what would that be? That would be 20 molecules per milliliter. I'd have what? I don't know. I don't want to do 100 milliliters of water. No, that would be 5. 80 milliliters. I'd have to add 80 milliliters of water to that cell. So if the cell has, 1 milliliter of water and 20 molecules, I add 70 more milliliters, and suddenly the concentration inside the cell is equal to the concentration outside the cell, just through movement of water. But to simplify it, I'm going to be talking about these tonicity solutions, isotonic, hypertonic, hypotonic, in terms of concentration of water inside the membrane versus outside the membrane. So in animal cells in an isotonic solution, they are healthy. But with plant cells in an isotonic solution, they are actually unhealthy. If you forget to water your plants in the day outside on the porch, when you get home, they're all droopy. And that's because the amount of water surrounding the plant cells and the roots has been used up over the day, and you're down to an isotonic concentration of water in the soil, right? And so the plant is all droopy. We call that, we refer to that as the plant cells being flaccid. All right. Hypertonic solution. In this case, the cell has more water inside the membrane than outside. So diffusion is the random movement of particles from areas of, that's right, high concentration to areas of, low concentration. So how is water going to move across that membrane? That's right, it's going to exit the membrane. So in a hypertonic solution, you might be referring to something as salt water, where in this case you might have a cell that salt can't pass the membrane. And so you put the cell in a solution that has a whole bunch of salt in it. And so there's a lot of salt in the water surrounding the cell. And thus, each of those salt molecules is taking up space that water could have taken up. And so you have less water surrounding the cell, a lower concentration of water surrounding the cell by default, because there's salt taking up space that could have been water. So, in this case, there's a higher concentration of water inside the cell versus outside the cell. So water is going to exit the cell. It diffuses across the membrane to the outside environment. So imagine your cell is a water balloon. What happens when you let water out? That's right. The cell will shrivel up. A water balloon shrinks, the cell is going to shrink and shrivel up. All right. So in a hypertonic solution, the cell shrivels up, and there's a fancy term for this in animal cells that's called crenation. So the cell shrivels up and usually dies. In plant cells, the plant is wilting. So the overall multicellular plant is wilting to the point of death. So this is when you come home and see it wilted and you add water and it doesn't perk back up. Those cells have all died due to losing water, and the term for a plant cell losing water is plasmolysis. We have a different term for a plant cell losing water because the cell wall around a plant cell doesn't change shape when the plant cell starts losing water. That cell wall is made of cellulose. It's strong. It won't lose its shape when water exits the cell. However, with less water in the cell, there's, the cell is still going to shrivel up. But in this case, it's the cell membrane shrinking away from the cell wall. So if this is my cell wall and here's my membrane, it's going to shrink away and shrivel up That's plasmolysis. In an animal cell, there's no cell wall, so it just goes and shrivels up. So here's an animal cell in a nice, healthy isotonic solution. And then we stick it in a hypertonic solution like salt water, and it shrivels up and dies. All right, next up is the hypotonic solution. In a hypotonic solution, there is going to be less water inside the cell than outside, so there's a lower concentration of water inside the cell than outside. You can also think about it as a larger concentration of solutes inside the cell versus outside. Remember, solutes take up space that could have been water. So whenever you add a solute, you decrease the concentration of water. The more solutes you have, the lower the concentration of water inside the cell. And so you get to a point where it's hypotonic. The concentration of water outside the cell is greater than the concentration of water inside the cell. So water is going to move from an area of high concentration to an area of low concentration. So it floods into the cell and swells that sucker up. And then if the cell swells up too much, if you fill a water balloon too much, it explodes. In animal cells, we call that lysing. Now in plant cells, something else entirely happens. And the reason is because plant cells have that strong cellulose cell wall. When you start to fill that sucker up with water, that cell wall is not going to break. That is strong. It will hold its structure, and that membrane can only move so far. And so the membrane can't swell up enough to break. And what you would get in result is that water enters the cell, and you get a pretty decent amount of pressure against the cell wall, but the cell stays intact, and so that cell becomes what we refer to as turgid. The plant cell is normal and healthy, and I just explained why. It's healthy because a plant cell with lots of pressure against the cell wall can stand against gravity more effectively. So, osmoregulation in cell walls. Turgor pressure is the pressure of water against cell walls. And that wall prevents lysing. So plants want to be turgid, so plants like hypotonic solutions. That's why you water them. Because turgid cells resist gravity a lot better than cells in isotonic solutions, flaccid cells, or cells in hypertonic solutions, which are plasmolysing and dying. So here's a nice little red blood cell, an animal cell, thrown into a hypotonic environment. And this is what it looked like at first. This is a healthy red blood cell, but then water floods into the cell and it swells up into this big balloon. And if you let it keep swelling up, well, eventually it's going to blow. It's going to lyse. You best check yourself before you lyse yourself. That's hilarious. Okay, so here's a nice comparison of animal cells in hypertonic, isotonic, and hypotonic solutions and plant cells in those same solutions. So here, animal cell shrivels up and plasmolyzes, or crenates. The animal cell shrivels up and crenates. The plant cell, the membrane, shrinks away from the cell wall. Membrane shrivels, and the plant cell is plasmolyzed, killing the plant cells. Here in an isotonic solution, we have a normal, healthy animal cell, but the plant cell is flaccid. It's not resisting gravity near as well because the concentration of water is sort of no net movement, and so your plant cell is sort of floppy. It's flaccid. And then finally, in the hypotonic solution, The animal cell is filled with water until it swells up so much that it can't swells no more, and it lyses. Whereas the plant cell swells up with water, and the cell membrane pushes up against the cell wall, and the cell wall holds its shape, and we have a turgid plant cell that resists gravity, and it's normal and healthy. So osmoregulation is the concept of water concentration regulation in a biological system. So maintaining a healthy concentration of water in the cell, keeping it from dying. So in an animal cell, you are preventing crenation or lysing. Contractile vacuoles can push water out of a cell. So that's a contractile vacuole. If you recall, chapter 4, we looked at a video of a contractile vacuole. You see that in a lot of protists, little single-celled organisms. When they enter into a freshwater concentration and they start to fill with water because it's hypotonic, their contractile vacuoles fill with water and then push the water out of the cell in order to keep the cell from swelling and lysing. Multicellular organisms will use organs such as kidneys. So our kidneys are responsible for getting rid of excess water or conserving water. So if I've drank plenty of water in a day, in order to keep my blood volume at a stable level, my kidneys are going to pull water out of my blood and fill up, and then I'm going to urinate, and it's going to be almost completely clear. Whereas if I haven't drank anything today, and and my blood volume is in danger of being low, my kidneys stop pulling water out of my blood, and they'll even pull excess water out of my urine and put it back into my blood in order to keep the blood concentration at a healthy level. And in that case, my urine's going to be like dark orange or amber and kind of like stinky if it's morning sometimes. Not that I know. Okay, moving on. You know what? This has gone on a while. So I better cut it here. Dang. I wanted it only to be 3 parts, but we're going to roll into a part 4. See you in the 4th video for transport proteins.